Good afternoon. This is the start of the afternoon session of day 2 of the IVRT IVPT workshop. This afternoon centers on IVRT Method Development, Validation, and Transfer. I'm happy to introduce the moderator of this afternoon, Tanaz Ramazanli. I refer you to her full CV in the booklet. Tanaz is a pharmacologist at FDA OGD. Just one element about some of her activities, in addition to review, is she also serves as a project officer for multiple regulatory science research initiatives related to development of bioequivalence standards for complex topical drug products. Tanaz, thank you for moderating this afternoon session.
Hi, everyone. Thank you very much, Dr. Pauly, for the kind introduction. It's my pleasure to start the afternoon session and introduce our first speaker, Dr. Theo Kapanadze. He is a co-founder and chief scientific officer of Diteba Research Laboratories Incorporation. He has more than 30 years of analytical R&D chemist and pharmaceutical industry experience. Theo, the podium is yours.
Okay. Thank you. Can you see my? Can you see my slides? Yeah. Okay. Okay. Thank you, Thomas, for introduction. And I want to back everyone to my presentation, in which I will focus on in vitro tested methodology, which is IVRT, that is using artificial synthetic membranes instead of biometric or ex vivo human scale. IVRT is used to monitor release diffusion of drug substances from semi-solid dosage forms, and as a tool to support product development has been around for approximately 50 years. Therefore, it's a relatively well-established methodology. IVRT mainly is utilized as a quality control measure to see release, detect changes made in a formulation or manufacturing process, lot-to-lot consistency, shelf life stability study, as well as the post-approval changes. In some cases, alone or combination with IVPT also could be considered as an alternative approach for bioequivalence assessment, generic versus reference drug. So again, IVRT mostly for those semi-solid formulations that apply to the scheme are well-established methodology and considered as ESP competitor. It has been many times discussed at the FDA workshops, meetings, literature guidance. However, there are still some challenging points triggered by either formulation specifics or application sites, which I'm going to discuss in a minute. Briefly, I'm going to outline rationales of IVRT, the key aspects of method development and validation in light of the discriminatory capability, and overall challenges using IVRT for shelf life stability studies and methods transfer. So since FDA posted product-specific guidance, PSG, first in 2012 and then 2016, with IVRT as bioequivalence option for topical acyclovir ointment and a cream, 15 to 17 in total generic products approved. Not bad. And briefly, in regards of theory of IVRT, 10 years ago, there was published an article which I like very much. It was for 50 years universally of Higuchi's classical equation, titled Higuchi's equation, derivation, application, use and misuse. They are very well presented mathematical models and framework within which theory is operating. Actually, there are highlighted 9 conditions limitations that originally has contemplated by Higuchi. The assumption has been made that the classical Higuchi's equation had a tremendous impact on the field of advanced drug delivery. This equation allows very easy calculate of the drug release from the complex type of the system. However, caution should be paid not to violate any condition derivation of equation is based on. The question is posed is Thus, as a draft document provides complete guidance on method development, validation, and data interpretation. Admittedly, for the first time, we got such a comprehensive guidance. This, of course, not SOPs, but quite complete emphasize all the key points. IVRT instrumentation, to which instruments are preferable, and how it could be qualified. IVRT method development, how to develop suitable methodology, what to include in MD report, and what needs to be submitted to regulatory agencies. IVRT method validation, so with key parameters have to be validated to prove how to pivotal study, design, and execute it. IVRT documentation required. Recently, there are standardized formats available to what data and how it has to be submitted. Now, briefly sum up IVRT rationale, the main principles. Drug release is controlled by diffusion and is not limited by solubility. Drug in a receptor medium, for example, saturated solubility of the drug receptor medium is high. Ideally, it should be 10 times, but sometimes 5 times it's okay. Yeah. The membrane does not act as a barrier, significant rate-limited factors, and does not interact with either application, products, or formulation solution. It should be inert toward both product apply and receptor. Diffusion coefficient of the drugs remains constant, steady approach. If plot cumulative amount API released versus square root of time has no linear path, then IVRT is not valid. Deviation from the linearity is usually observed at later time point. It's majority, but in reality, deviation from the linearity can happen any time point. The initial steps in IVRT methods development is choice appropriate diffusion system. There are presented examples of the in vitro release test systems proposed for industry with respect to reproducibility and discriminatory capability. Static French diffusion cell, this is the first choice. Historically, this system has been successfully used for IVRT method development, particularly for dermatological products. USB aperture 2 with immersion cells and USB aperture 4 with the semi-solid adapters considered its operational principles. Very rare cases could be used for dermatological products. It might better approach for ophthalmics or buccal sublingual products. The majority of the APIs absorbs into the systemic circulation. There is full automated testing as an example. Let's say the Phoenix from Telida. There are other manufacturers as well. Logan, Thermigir, for instance. Automated systems are obviously preferable for IVRT studies to their advantage over the manual operation in both technically handling the experiment and compliance viewpoint. For example, validated software capable digitally storing protected information for testing conditions such as temperature, mixing speed, sampling time point, volume, with the audit trail, of course, and all could be presented to the regulatory bodies upon request, and useful for method transfer due to easy to reproduce standardized conditions in the operational protocol. The selection of the optimal IVRT conditions starts with choice of the receptor solution. This already discussed. Rationale for a selection of receptor medium is based on the solubility and stability of the drug acting in a medium. The receptor medium must provide adequate stability and sync condition. Ideally, receptor medium could be a buffer solution. However, the products formulated with water-insoluble drugs, the selection of appropriate receptor medium to maintain sync characteristics and it is a challenge. And in order to facilitate and monitor drug release from such topical formulation, it might be necessary to alter receptor fluid pH or use surfactant or use non-aqueous medium in which the drug is more soluble. Introducing the receptor solution, alcohols, propylene glycols, for instance, is adaptable. In any ways, IVRT is not expected to correlate with in vivo data. One practical consideration to choose a receiving medium is that allows sufficient amount of the active ingredient released within a reasonable time period. Normally, we start with 3 different receptor solutions and 3 different membranes. The main consideration, besides solubility, are good linearity, are square more than 97, as they discussed, or higher, and smaller error bars, cell-to-cell variability. The next is the membrane. Commercially available synthetic Vitro membranes with different pore size could be evaluated for their inertness toward both the receptor medium and formulation. Membrane should be demonstrate no binding to the drug. Some examples are listed here: the nitrocellulose, duraport, capri, nylon. Dialysis membranes, rare cases, finding application for some suspensions, emulsions, or liposome encapsulated actives inevitably introduce rate-limited diffusion. The first step, visual examination prior soaking membrane in the release medium for at least 30 minutes and before dosing. The membrane should not exceed significant changes in appearance. The next is the conduct membrane binding test. Normally 3 replicates, as mentioned today, with the quality control, standards, low and high quality control replicate. The immersed membranes is the same volume, let's say 10 milliliter of cells volume. Membrane should be soaked for a period equal or greater of the time length of the IVRT experiment, let's say 6 hours. The incubation temperature 32 or 34 if you go for scenario. Also, control standards should be evaluated without membranes, should be kept under the same storage condition. Further, analyzing concentration standard solution with membranes and without membranes, and they calculate the recovery. They have ±5% of acceptance criterion that will validate the membrane. The matrix-specific, such as rheological properties, must be taken in consideration when developing appropriate IVRT method. For example, for most fast-release matrix, early sampling time point between 0 to 3 hours could be find it more discriminative. For example, quality indicating the later time point. Good examples are propylene glycol or polyethylene glycol-based ointment. Both are unaccountably miscible with aqueous and many of organic solvents. So due to fast solubility, formulation matrix migrates through the membrane into the receptor medium and fast release the drug. There is fat chance you find such solvent or mixture of solvents that immerse membrane and matrix products but accept the predominantly API. That's a huge famous equation based on the ointment film. No swelling, no ointment matrix dissolution. So we have ESP 7024 specifies VRT duration for 4 to 6 hours. So an IVRT with a slope shorter than 4 hours duration inevitably could trigger observation from the reviewer. Needs a justification. Another example is a very slow release matrix like petrolatum-based ointment. VAX does not want to release drugs even after 48 hours in many of traditional IVRT receptor solutions. There is also an issue such as compatibility of the matrix with the receptor medium. Hydrophilic solvents clog the matrix and after a certain time point, release no release, stop the drug release at all. Needs to use more harsh organic solvents, for example, THF, with the optimal mixture of polar solution to help to dissolve the drug that could be the hydrophilic. So in vitro release rate testing duration and sampling time point may be vary, depends on formulation matrix. Therefore, the IVRT duration, length of experience in many cases is the subject of the discussion. EMA has already introduced in the latest guidance, IVRT duration should be sufficient to characterize release profile more than 70% of API release. Apparently, this is more appropriate, for example, again, ophthalmics or nasal inhalable, buccal sublingual site absorption. Most of topical dermatology products do not reach 70% release, 6-8 hours IVRT duration. So to achieve this value, prolonged experiments is needed that do not mimic in vivo conditions. So it is clear that at least 5 time points, better to use the seed, must be used for a linear regression path. Since Higuchi's model proposed to use pseudo-infinite dosage model, requiring a high initial access of the drug on the sampling compartment in order to comply with a pseudo-steady state approach, a direct proportionality between the cumulative amount of drug release and square root of the time. Higuchi's model is square root of time kinetics. Prediction is to expect linear part of release for the first 60% of total drug release. So if the slope does not deviate from the linearity, then limiting the method by the amount API release, let's say by 30% or less, I think makes no sense. It is important applied dose amount to be accurately dosed. Reproducibility plus minus 5% EMA has already proposed in the guidance. Quite tight, but it is important for mass balance dose depletion calculation. During dosage, it is critical to avoid bubble creation. Therefore, method of product application and sample and volume collection has to be standardized and validated. Starting time point must be staggered and synchronized with the sampling time for its successive diffusion cells. FDA guidance recommended keep cells occluded, no formulation, evaporation, or leak. At Diteba, we're using accurately weighing sample before apply. This is an example. First, we allocate the samples directly from the tube to the large syringe, and then the 1 milliliter syringe. This depends on the product. Predeterminate volume, different from different products, loading in 1 milliliter individual syringes. Then accurately weigh and recording, and then apply to each membrane into the dosage rings and closing with the glass tops and clamp. No evaporation or leak. The application of the samples on 6 cells takes less than 1 minute to chemist. Starting time, synchronously mixing all 6 cells, and time elapsed. Weighing empty syringe and recording the exact amount of product applied to each individual cell. That helps to accurately calculate the mass balance dose depletion. Out of it, the major consideration for developing an IVRT method is the discriminative power. Manufacturing methods and process may change formulation attributes and affect the rate of drug release and drug bioavailability. IVRT method should be capable to discriminate the effect of process variability in the production of the test formulation, as well as distinguish variability of compositions that may impact in vivo performance. So during validation, method routinely challenge sensitivity, specificity, and selectivity. IVRT method sensitivity and selectivity determines effects of changes concentration in API. Expectation is to demonstrate proportionality of changes in the release rates as a function of drug concentration in a formulation. Through the specificity, the expectation is to have good linear response with the amount API change. This linear correlation is very true for aciclovir and the ointment PHG was originally proposed with, and many other dermatological products. No problem at all. However, in some cases, when the product matrix API is presented at saturation level, API either dissolved or suspended at saturation level, therefore, any efforts to add more API leads to the precipitation in the matrix. We had a case when making even 120% liver claim was challenging without breaking the matrix. Under microscope, you can see the lumps, large API aggregations. Such products normally is giving the same or even less slope than RLD with high cell-to-cell variability due to uncontrolled release. That leads failure IVRT lots comparison test to demonstrate methods discriminatory power on 100% liver claim versus 115. In such cases, one possible suggestion could be proposed. Make series altered lots, 50%, 75%, 100%, and 125%, and demonstrate trends of increases, decreases slope as a function of API concentration. Perhaps makes sense to narrow the confidence interval, for example, as EMA proposed 90% to 110% instead of 75 or 133 confidence interval. However, since restrictive confidence interval might compromise lots during the pivotal study. So this is the issue to be considered when you tight acceptance range. Case number 2. Some products with slow or well-controlled release matrix are close to the classical field, which is model ointment films. At the increased level of API concentration might not necessarily demonstrate proportionality in release rate increase. Twice increases in concentration in 100% labor claim would not always result double slope, but increases is less than 2. In many cases, following like square root of 2. Double increases in concentration shows only 1.4 increases in slope, as predicted by classical equation. Accordingly, the Wixon rank sum man witness rank test at 90% confidence interval and altered lots with 150% labor claim could easily fall between 0.75 and 1.33 in this wide range proposed by USP. Normally, such formulation only the lot with 200 or more labor claim are falling outside the specified range, 75 to 133. It is not because the method is not sensitive, but nature of the diffusion process. For some variables, it is slow, well controlled. IVRT method has been optimized with all feasible conditions, but still 150% labor claim within 75 and 133 range. Suggestion, again, make series of altered load with the API concentration at various of level, for example, 50%, 75%, 100%, 125, 150, 175, it can go to 200 to 225 or more if you may. And demonstrate the trend, there's a lot with the low API contents are displayed as a low release rate and lots with a high API, with a high slope. It is a high slope. And importantly, this variability must be larger than method reproducibility, intraday precision, outside of error bar. Currently, there is a need for alternative statistical approaches that could be applied for such cases to demonstrate significant differences among the lots. For example, our statistician partner company suggests that to use more than 6 cells, for example, 10 cells, it would help to do more fair lot comparison, improve IVRT methods sensitivity. It is under investigation. The case number 3 in this very rare case is that API is in a formulation acting as a viscosity builder. Consequently, changes made in the amount of API will inevitably affect the rheology of the product. The viscosity of altered lot with a 50% and the 100% labor claim are very different from the reference product. Subsequently, the release rates not adequately responded to the changes made in the amount of API. The adjustment of the viscosity within Q1 and Q2 requirement equivalency is the challenge. 2 possible suggestions here. Introduce the additional viscosity adjustment ingredient, which is not in R&D, in a formulation with 50% and the 150% Lipaglyn, if regulatory agency, of course, agree. Or since API cannot be changed, altered lot could be done by variation of amount of inactive ingredient. Methods well responded to the changes made in amounts of inactive ingredients, so-called supplementary selectivity. Now, briefly, I would like to highlight some of the critical points relating to the stability program and IVRT method transfer. One additional challenge is that inclusion of IVRT method in shelf life stability program for a topical product. It is necessary to consider the inherent IVRT methods variability. The source IVRT variability, as we discussed, can cause complex of factors such as bubbles entrapment during the product application. Bubble between membrane and products could be created due to back diffusion. Membrane qualities might slightly change during the manufacturing product long-term stability study. During aging, semi-solid products could experience metamorphosis. Therefore, in many cases, regardless, IVRT method successfully has been validated within 10 or 15% slope variability, but a routine-based stability program could observe a larger inconsistency in release rate. In order to propose practical applicability as desired, it is critical to establish more realistic criteria for each product. It is also suggested, it is important, in each time point during the IVRT run, include reference load as a positive control to make correct judgments when abnormal slope observed that not related testing error. About method transfer, as we know, until now, there is no guidance for a IVRT method transfer, therefore no standard protocol available. It is crucial for a release rate reproducibility. Both sides have exactly the same diffusion system, preferably an automatic, the same manufacturer. Easy to reproduce diffusion parameters, cell dimensions, volume, mixing rates, method transfer protocol could be designed as follows: 3 products in 3 different ways. Negative control, which is altered formulation. Positive control could be the R&D and test formulation. The both sides of the IVRT data should meet their pass criterion. A difference between slope, testing slope should be the less than 50 or 10%. So in conclusion, determination of the in vitro related profiles is useful in many scenarios. During product development, it can provide essential data of product's microstructure, valuable to optimize both formulation and production process. On the other hand, during the late stage development, as well as a post-marketing phase, as a quality control tool to monitor batch-to-batch consistency. In many advantages, the main advantage of this method rely on its high sensitivity and discriminatory power. In specific conditions, in vitro release testing can be used to demonstrate products bioequivalency when compared to pre-approved products. Regulatory bodies are increasingly expected IVRT to be used as a quality tool as a part of all semi-solid QC release and stability specification. Traditionally used vertical diffusion cells, that testing is now been automated to improve throughput and reproducibility. And here is Diteba Laboratories via IVRT business since 2005 with numerous successful NDI submissions. And thank you for your attention.